Hi, my name is Jordan Avery. The regulation and coordination of the GI tract during the gastric phase of digestion relies on integrated signaling between neural, hormonal, and paracrine pathways to regulate gastric secretions, motility, protective mechanisms, and downstream digestive processes. The enteric nervous system, composed of the submucosal and myenteric plexuses, has its own short reflexes that functions autonomously to regulate GI activities. Sensory neurons detect mechanical and chemical signals, while motor neurons control motility, secretions, and endocrine functions. ENS-driven reflexes, such as peristalsis, can operate without input from the CNS. Neurotransmitters like acetylcholine and substance P enhance functions, while nitric oxide and vasoactive intestinal peptide act as inhibitors. Gastric activity is largely modulated by parasympathetic input from the vagus nerve, with sympathetic input acting as an inhibitor. The vagovagal reflex, involving both afferent and efferent fibers, regulates gastric secretions and motility. Vagal afferent fibers detect gastric distension and chemical signals, and efferent fibers complete the reflex circuit with the release of acetylcholine, stimulating gastric acid secretion and motility. Regulation in the cascade of events involves several distinct cell types that perform specialized functions in regulating secretions. These cells are gastric cells, enterochromaffin-like cells, parietal cells, and chief cells. Mucous neck cells and epithelial cells perform protective mechanisms, while delta cells, not seen here, produce an acid inhibitor. In the gastric ostitic glands of the stomach, parietal cells secrete hydrochloric acid. Two primary pathways stimulate its release: acetylcholine binding to M3 muscarinic receptors and the hormone gastrin binding to CCKB receptors. Gastrin is released by G cells located in the stomach's antrum into systemic circulation in response to vagal stimulation and gastrin-releasing peptide. Gastrin binds to enterochromaffin-like cells, releasing histamine, which acts as an amplifying paracrine signal by binding to H2 receptors on parietal cells to potentiate the actions of acetylcholine and gastrin in acid secretion. Intrinsic factor, also released by parietal cells, is essential for vitamin B12 absorption. A locally acidic environment is necessary for killing bacteria, inactivating salivary amylase secreted during the cephalic phase, and inactivating protein digestion. Acetylcholine triggers the release of inactive zygomin pepsinogen and gastric lipase from chief cells. These enzymes are important for protein and triglyceride digestion. This is done via exocytosis due to increased levels of intracellular calcium. Conformational changes due to protonation shifts the inactive zygomin to reveal an autocatalytic cleavage site. Pepsinogen self-cleaves to fully activate pepsin. Pepsin acts on remaining pepsinogen in a positive feedback loop. Acid also serves the purpose of denaturing digested proteins, increasing their surface area to allow pepsin to act. Mucous neck cells and epithelial cells enact protective mechanisms to prevent damage to the stomach. Epithelial cells secrete a thick alkaline mucus layer that coats the stomach lining, acting as a barrier from low pH levels. Bicarbonate ions are secreted by mucous neck cells, which neutralizes excess acid, preventing ulcerative damage. The stomach maintains pH balance through negative feedback. When the pH drops below 3, G cells respond by reducing gastrin secretion and decreasing acid production. Somatostatin, released by delta cells in the gastric gland, further inhibits acid secretion and reduces digestive activity as the pH decreases, preventing excessive acidity of chyme as digestion progresses. Receptive relaxation is a vagovagal reflex, acting to reduce pressure and increase the volume of the ORAS stomach. Nitric oxide and vasoactive intestinal peptide mediate the smooth muscle relaxation and expansion to hold food. Without this regulatory feature, the small intestine wouldn't be able to digest and absorb the chyme properly. Peristalsis contractions occur in the fundus and body. It is a propulsive movement that pushes the chyme forward into the intestinal phase, mediated by the enteric nervous system. Hydrochloric acid and pepsin are mechanically mixed to increase nutrient surface area. Movement is towards the pyloric sphincter, and things that are important in regulating this are acetylcholine, substance P, nitric oxide, and vasoactive intestinal peptide. Gastric emptying is regulated by two main hormones in response to increased fat content and acidity. Cholecystokinin is released by eye cells in the small intestine and slows gastric emptying when lipid levels are high to optimize digestion. To allow for the neutralization of acidic chyme by pancreatic bicarbonate, secretin is released by S cells in the duodenum to slow gastric emptying. Insufficient acid production, called hypochlorhydria, impairs protein digestion, leading to malabsorption and nutritional deficiencies. Bacterial overgrowth of Helicobacter pylori is an example of a bacteria that causes low acid production. This damages the stomach lining while also allowing the bacteria to thrive. Excess acid production can result in gastritis, acid reflux, gastroparesis, and peptic ulcers. A rare disorder this relates to is called Zollinger-Ellison syndrome. It is the result of gastrin-releasing tumors causing excessive acid secretion and ulcer formation. Disruption in neural signals or hormonal imbalances can lead to these disorders. For example, chronic stress can increase vagal activity, exacerbating acid secretion. Two common treatments for excessive stomach acid include medications such as proton pump inhibitors, an example would be omeprazole, and H2 antagonists such as cimetidine. Proton pump inhibitors, also known as PPIs, work by reversibly inhibiting the proton pump on the luminal surface of parietal cells in the stomach. H2 blockers work by competitively binding to histamine H2 receptors on parietal cells, preventing histamine from binding, which reduces acid production. Some of the consequences of these are listed. Some negative effects could be reduced acidification, which could inhibit pepsinogen activation and gastric protein digestion. Reduced acidity may decrease the effectiveness of the mucosal protective barrier. Long-term use of these drugs can lead to bacterial overgrowth and an increased risk of gastric infections. Increased sensitivity to gastrin can occur. G cells will release more gastrin without the inhibition of low pH. Elevation in gastrin triggers hypertrophy and hyperplasia of parietal cells, leading to an exaggerated response to acid and greater likelihood for ulcers to occur upon ceasing to take the drug. The gastric phase of digestion is finally regulated by a combination of neural, hormonal, and paracrine mechanisms. The enteric nervous system autonomously controls motility and secretion, while the vagus nerve modulates these processes under parasympathetic control. Gastric acid secretion and enzyme release are tightly controlled by cellular signaling, and protective mechanisms prevent damage from excessive acidity. Dysregulation of these processes can lead to a range of digestive disorders, highlighting the importance of this coordinated system for optimal digestion and gut health.